Good morning, everyone. So my name is Yanyan Zhang. So I'm from civil engineering at NMSU. So today, Dr. Zhang Xu from biology and Dr. Ryan Ashley from animal science will give some aid, and I, of course, will give some update on the toxicity assessment on treated produced water. So in addition to conducting the conventional stand like a whole effluent toxicity assay like fetaminyl, ceridophenadubin, so we also did other in-depth analysis to evaluate the risk of treated produced water. So in my lab, I was using the human cell line, see whether there is potential human risk by using the treated produced water. So I'm also using the zebrafish for the long-term experiment, in addition to the previous embryo experiment study we did. So we share 70% gene with the zebrafish. So that's why this is a very good model to understand the human risk. So after the human cell line zebrafish study, so Dr. Zhang Xu we'll talk about the recent mice study. We did both acute and subchronic study starting this January. So we already generate a good amount of result. And after that, Dr. Ryan Ashley, he is an expert in animal reproduction. He will talk about the study plan, research plan on the impact of treated produced water on mice reproduction. So for human cell line study, we use 4 types of human cell line, including breast cancer cell lines, kidney cell lines, liver cell lines, and lung cell lines. So we did basic cell viability study by using 2 types of assays. One is MTT assay, which is enzyme-based assay. So the other one is cell title, GLUE, for which is based on the ATP of the cells. So after that, we also see whether there is activation of hydrocarbon receptor. So this is based on the gene expression study. So the bad thing of activation of hydrocarbon receptor is it will potentially generate some toxic metabolites, and also it will interference with overall gene expression of the cells. So we also test whether the treated produced water will cause some estrogenic activity by doing the gene expression study of the CYP19A1, and also we will check whether there is any inflammation response by checking the gene expression of the HMOX1. So due to the time limit, I'm just giving you a summary of the result instead of showing the real data for each assay. So those is a viability result. So we can we compare the 2 types of system. One is thermal-based system. The other one is membrane-based system. So for the raw produced water, we can tell both type of the viability assays showing. So the raw produced water is very toxic, either showing the 0% viability or 10% viability. However, as long as we do the desalination for the thermal distillate and our permit, there is no impact on viability. That's why we see 100% viability after those treatment. So however, the viability study is not that sensitive for some compound. As we know in the thermal distillate, even some our permit, we still see some compounds, especially organic compounds, ammonia in those things. So that's why we are seeing some in depth. So that's why we did the activation of AHR. So that means normally as long as we have the aromatic compounds, so they will show the response. So we will have the upregulated gene expression of the CYP1A1 or CYP1B1. So as you can see, for the distillate, thermal distillate, since we still have the residual organic compounds, so we did see the activation of the AHR. And but for our process, so after membrane process, our permit, there is no activation of the AHR. So for the estrogenic activity, we only see the induced estrogenic activity in the raw produced water, so not in the thermal distillate, not in the alpermate, not in the final effluent. So let's do a comparison with the standard whole effluent toxicity assays. So those are the 4 types of pilot scale treatment. So 3 of them are fine, except 1. So thermal process 2, so this is a process we see a high TOC in the effluent in both distillate and also in the final effluent even after pollution step. So we see about 20 to 30 milligram per liter of methanol and some other trace organic compounds. So you see so they didn't pass the red test. So that means this is the table for the no observed effect concentration. So that means we use 100% water. So we did see the inhibition of those organisms by using 100% water for thermal process number 2. So for only the 50%, if we dilute the water by 1 time, so 50% water showing no impact. So that means a traditional standard wet test still very good indicator as a screening tool for the treated produced water. So next, I want to give you some result for our long-term zebrafish explorer study. So for the conventional wet test, normally the chronic test is 7 days, but we don't really know. So if we let the organism stay in that water for much longer time, what will happen? So that's why we use the zebrafish, the long-term experiment. So for this study, we use the adult fish, so which is about 8 months old. So we put 20 fish for 1 water sample, including 10 female and 10 male. So we put every 5 fish in those 2-liter beaker. So that beaker has 1.5 liter water. So we have the 3 types of water. So control water is the NMSU tap water after the aquarium water filtration system. So this system has the reverse osmosis followed by ion exchange process. So for the treated produced water, we use the membrane effluent. So those are the basic water quality parameters. As you can see, the TDS is very low, TOC is just above detection limit. Ammonia is below the detection limit. So another water we're using is 400 times diluted raw produced water. So that's kind of scenario if we have the spill, if something happened, worst case scenario. So how we can do the remediation? So can we just do the dilution water by using 400 times the fresh water to dilute it? So after dilution, the TDS still meets the drinking water standard. So TOC is below the detection limit. The only thing is the ammonia, still 1.45. So which still it's kind of toxic level. The pH of water is about 7.5. So it gives us the free ammonia level of 0.02 milligram per liter. So some fishes, aquatic organisms, still sensitive to that one. So all the water are remineralized with the EPA moderately hard water recipe. If we don't really want to use the deionized water to maintain the fish. So and also we add some aquarium salts to maintain the appropriate conductivity level. So we did the 28 days experiment. So we change the water every 2 days. So those are some result based on the liver biomarker. So the first biomarker is superoxide dismutase. So this is enzyme can convert the reactive oxygen species to hydrogen peroxide. And then the hydrogen peroxide will be, will be degraded by another enzyme, which is the catalyst. So they can convert the hydrogen peroxide to the non-toxic species. So that's the 2 enzyme analysis. So both increased level, decreased level are not good. So why are using those oxidative stress? So because many compounds, especially the toxic compounds, can cause the oxidative damage. So that means they will produce the reactive oxidative reactive oxygen radicals. So as you can see, the SOD. So there is no difference among those three waters. So the second one is the catalyst. This is the enzyme convert hydrogen peroxide to the non-toxic species. So we see the decreasing in the 400 times diluted raw produced water. So decreasing is even worse than increasing. So that means that the overwhelming oxidative stress caused by diluted raw produced water. So there are some enzyme inhibition in this case, but treated produced water, there is no difference with control water. The last one, which is more important, so that's something called malone dialdehyde acid. So this is key byproducts of the lipid peroxidation. So lipid in our cell membrane. So if we have the oxidative stress, the lipid will be damaged, and then they will have these key products, which is MDA. So if you see increased level of the MDA, means definitely there is a cell membrane damage. So for our control water, which has actually similar, sometimes even higher level than treated water. But for the raw produced water, diluted raw produced water, which has highest level. So those are the male, those are female. So that means definitely 400 times diluted raw produced water are causing the liver damage. So interestingly, if we compare the control with treated produced water, you see more damage for the control compared with the treated produced water. So that means it seems treated produced water sometimes cleaner than the control water. For the control water, we already use the best available water we can get. But sometimes, so we cannot tell which one is cleaner, maybe. We're still doing some ongoing work, checking the liver histology and see whether there is any damaged liver cells. We are doing the RNA-seq to understand the whole metabolic pathway affected by the different type of water. So this is a summary. For the human cell line study, we found the polished thermal distillate or our permit has no impact on those cell availability. So only some thermal distillate has residual organics which will activate some AHRs due to the presence of aromatic compounds. Some of them may be unknown compounds. And the polished water, there is no cytotoxicity, no estrogenic activities, no activation of AHR. So for the zebrafish, the treated produced water has no impact on the fish survival. I didn't say it, I forgot to say it, all the fish survived after those 28 days, even for the diluted raw produced water. And the treated produced water didn't result any oxidative stress. Only the raw produced water caused some oxidative stress. So I still have some ongoing study to further understand the result. So next step work, we are going to use some solid phase extraction to understand the cumulative impact on the human cell lines. Right now we're only doing the 24-hour exposure, so maybe not long enough, the concentration maybe not high enough in the treated produced water. So that's why we want to concentrate the compound by using the solid phase extraction. Another thing is previously we only used the AHR, which is the aryl hydrocarbon receptor, which only can recognize the aromatic compounds. So we want to use other nuclear receptors which can serve as the xenobiotics sensor. So we have different kind of receptors can sense the, for example, a hormone, steroid, and the pharmaceuticals and all other compounds which causing the problem will give us a better understanding what are the residual compound in the treated water. So for the zebrafish study, right now we're also using the juvenile zebrafish and see whether the treated produced water will affect the development of those fish. So we will do the reproduction study. We will check the numbers and quality of embryos. We will do some histology work for the reproduction organ, ovarial and the testicle. So we will also do something like called vitellogenin, so which is a very important compound for the egg yolk, so which is important for the egg development and also for the reproduction organ development. So that's everything from me. I want to thank the student Pyumi, who is sitting here doing the human cell line study, and we have the Tashan on the back who did the zebrafish study. So after that, I'm giving the floor to Dr. Zhang from biology, talk about the mice study we just did in the last few months. Thank you.